CD3 bispecific antibodies hold great promise in the field of cancer therapy. Today we will talk about CD3 bispecifics and introduce a powerful tool for advancing CD3 bispecifics preclinical evaluation. We will start with the introduction part of CD3 bispecifics, which is also called T-cell engager. According to a recent article published in Nature Reviews, there are over 200 different bispecific molecules involved in more than 300 clinical trials. And 50% of these bispecifics are either in phase 2 or phase 3 or have already been approved. Approximately 3 out of 4 bispecifics are being developed to treat solid tumors, and this area is largely dominated by T-cell engagers, which make up about 33%. And on the other hand, for hematological malignancies, T-cell engagers are even more prevalent, making up 75%. From a market perspective, the bispecific market was valued at around $70.8 billion in 2022. Looking forward, this market is expected to grow very fast and highlights the expanding role and potential of bispecific antibodies, especially CD3 T-cell engagers, in cancer treatment. Until May 2024, 15 bispecific antibodies have been approved, with 12 of them being used for cancer treatment, and 10 are classified as T-cell engagers. This clearly highlights the development of T-cell engager bispecific antibodies has become a mainstream focus. Bispecific antibodies are available in different formats, address different targets, and the different anti-cancer function via different molecular mechanisms. There are the formats of approved T-cell engagers. We won't go into too much detail here. This is the mode of action of CD3 bispecifics. Briefly, it simultaneously binds to tumor-associated antigens on tumor cells and CD3 on T cells, cross-linking these two cell types together to form synapse. And then the synapse promote T-cell activation, leading to the secretion of inflammatory cytokines and to kill the tumor cells. They also recruit immune cells into the solid tumor environment. And let's now take a look at some representative drugs. First is a CD3 EpCAM antibody. It is the first bispecific antibody to be approved for clinical use in 2009. Many types of cancer cells carry EpCAM on their surface. It binds to cancer cells, recruit T cells, and engage antigen-presenting cells, such as macrophages and dendritic cells, so it is so-called tri-functional antibody. The activated T cells and macrophages, DC, and natural killer cells will destroy the cancer cells. By reducing the cancer cell number in the abdominal cavity, it will help alleviate malignant ascites. However, in 2013, this drug was voluntarily withdrawn from US market and then EU market for commercial reasons. This is a phase 2 or 3 study involved patients with malignant ascites caused by different epithelial carcinomas. Cetuximab showed benefits regardless of the primary tumor metastasis or age, and extending the median puncture-free time to 77 days, and thus avoiding about 5 paracentesis procedures. In human anti-mouse antibody and human anti-rat antibody responses were noted due to its chimeric nature, but no safety pattern changes were reported after the last infusion. And because of the unspecific Fc gamma receptor and T cell receptor activation due to retained Fc gamma receptor binding, the next generation T cell engager were designed without an Fc portion to improve safety and efficacy. In 2014, Amgen's Blincyto became the second FDA-approved bispecific T-cell engager, also this format called BiTE. And this is an anti-CD3 CD19 BiTE antibody that links CD3 T cells and CD19 B cells, leading to T-cell mediated killing of B cells. It targets CD19 and overcoming B-cell leukemia and lymphoma. The T-cell recruitment and subsequent activation only occurs when the second arm of the BiTE antibody is bound to its target antigen on the tumor cell surfaces, and so it's ensuring the specificity. Because of lacking of Fc region, the drug has no ADCC or CDC function and has a very short half-life of 2 to 3 hours, so it's requiring the continuous infusion. And about 98.5% of patients experience side effects, with the most significant being neurotoxicity and cytokine release syndrome. One of the critical issues considered in blinatumomab therapy is the risk of resistance. As the table shows, one possible mechanism of resistance is the loss of the target antigen, and targeting mixed lineage leukemia rearranged BLL patients with the blinatumomab is another possible mechanism of blinatumomab resistance, which may switch lineage from ALL to AML. Increased expression of T-cell exhaustion markers like PD-1 and PD-L1 was observed following the therapy, so the immune checkpoint inhibitor may influence the drug efficacy. The blockade of the PD-1, PD-L1 pathway or CTLA-4 could enhance the T-cell function and improve the drug efficacy in patients who expressed T-cell exhaustion markers. Currently, several clinical trials were conducted to explore the effectiveness of combination therapy by blinatumomab and the immune checkpoint inhibitor in relapsed and refractory ALL patients. CD3/CD20 is currently one of the most competitive bispecific antibody target combinations, and this drug is co-developed by Genmab and AbbVie for the treatment of mature B-cell non-Hodgkin lymphoma subtypes. It received its first and conditional approval on May 2023 in the USA for the treatment of adult patients with relapsed or refractory DLBCL. In the preclinical evaluation, the drug resulted in potent and selective T-cell mediated cytotoxicity against CD20 positive B cells, and its IgG1 region is designed to be inert to avoid triggering unwanted immune responses such as ADCC or CDC. The drug is developed using the dual-body platform. This platform utilizes a natural Fab-arm exchange mechanism to combine two different antibodies into one and creating a bispecific antibody that can simultaneously bind to any two different targets. Single-agent treatment previously demonstrated potent antitumor activity in dose escalation across B-cell non-Hodgkin lymphoma subtypes. And in the pivotal phase 2 clinical studies, sub-Q monotherapy demonstrated response in relapsed or refractory DLBCL patients who have received at least 2 prior treatments. And here, the overall response was seen in 61% of patients, and 33% received complete remission. The drug can cause serious side effects, including CRS and immune effector cell-associated neurotoxicity syndrome. And sub-Q administration resulted in deep and durable responses and manageable safety in highly refractory patients. On June 2023, FDA granted accelerated approval of glofitamab for relapsed or refractory DLBCL. The drug is a novel IgG1-like humanized bispecific CD3/CD20 antibody with a Fc silence modification. It has two CD20 binding sites and one CD3 binding site. This unique 2-to-1 structure enhances the activity of CD20 T-cell engager, and compared to traditional 1-to-1 bispecific antibodies, this will resulting in a stronger target binding capacity and efficacy, and allow for more potent T-cell killing and lysis at low antibody concentration. And this is a phase 1 or 2 dose escalation and expansion study. In the phase 2 part, patients with relapsed or refractory DLBCL who had received at least 2 prior lines of therapy were enrolled. And after pre-treatment, patients received 12 cycles of treatment based on a step-up dosing strategy. Most patients were elderly with advanced diseases, and among them 33 had received CAR-T therapy, and the majority were refractory. The results showed at 12 months PFS rate of 37%. Here are the results of extended follow-up for the key phase 2 expansion cohort. With a median follow-up of 18.2 months, the results showed that 40% of patients achieved a complete response, and 52 had an overall response. Around 70% of patients are still in remission at 18 months, and the overall survival rate at 18 months was 41%. These findings highlight that glofitamab, with its fixed treatment duration, can achieve early and long-lasting responses in patients with relapsed or refractory DLBCL. And importantly, no new serious side effects were found during the extended follow-up. CRS remains the most common side effects occurring in most of the patients, but most cases are mild or moderate. The safety profile was consistent with earlier reports, and no new severe side effects linked to glofitamab. Next, let's look at the overall landscape and challenges of CD3 bispecific therapy. Since June 2022, the FDA approved 8 CD3 bispecific antibodies in succession. Prior to this, only blinatumomab, approved in 2014, was on the market. And this makes the transition of the overall technology into the industrialization and commercialization phase. Many other CD3 bispecifics are currently in clinical trials targeting well-established B-cell markers like CD19, CD20, CD38, and BCMA, and the myeloid markers like CD33 and CD123. And currently, T-cell engager antibody technology is expanding from blood tumor to solid tumors on a large scale. Rapid clinical progress, including the CD3 DLL3 bispecific antibodies focusing on small cell lung cancer indications. And many companies have venturing into the solid tumor field. The tumor-associated antigen like CEA, EGFR, EPCAM, HER2, and PCMA are all the hot targets in clinical studies. These are targets of T-cell engagers in the early phase of clinical evaluation in solid tumors. We can find DLL3, CEA, PSMA, which are hot targets in clinical and preclinical research. It has been reported that cytokine release syndrome and the immune effector-associated neurotoxicity syndrome are main adverse events. CRS can be induced by direct target cell lysis with consecutive release of cytokines like interferon gamma or TNF-alpha, or by activation of T cells due to therapeutic stimuli with subsequent cytokine release. And these cytokines trigger a chain reaction due to the activation of innate immune cells, such as macrophages and endothelial cells, with further cytokine release. Other less common but important side effects including hepatotoxicity and cardiac toxicity. In the table listed, the toxicity described for solid tumor T-cell engager therapies. Compared to blood tumors, solid tumors generally respond less effectively to T-cell engagers. Here listed three main hurdles for CD3 bispecific therapy in solid tumor. The first one is that the antibody will bind to tumor-associated antigen expressed on healthy cells but not in tumor cells, and thereby redirecting the T cells towards normal tissues. This will result in permanent tissue destruction. And the second reason is that in solid tumor, there is sparse or no T cell infiltration, and thereby preventing the CD3 bispecifics from cross-linking T cells to tumor cells. So the treatment efficacy is limited. The third reason is that in solid tumor microenvironment, there are multiple immunosuppressive cells, including cancer-associated fibroblasts, regulatory T cells, and tumor-associated macrophages. These cells will hamper the quality of effector T cells. Furthermore, the upregulation of immune checkpoints, such as PD-1, CTLA-4, will further decrease the function of tumor infiltration lymphocytes. To improve tumor selectivity and specificity, several effective strategies can be employed. First, target tumor-associated antigens that are exclusively expressed by tumor cells to ensure the CD3 bispecifics specifically target cancer cells without affecting normal cells. And second, increase the tumor-associated antigen avidity by generating T-cell engager with a 2:1 ratio, like the 2:1 CD3 bispecific antibodies, which have two binding arms for the tumor antigen and one for the CD3 to enhance the specificity and effectiveness. Third is to mask the binding arms of CD3 bispecifics using a protease cleavable linker, making them only active inside the tumor environment where specific proteases are presented, and thereby to reduce off-target effects. The fourth is to ensure local delivery of CD3 bispecifics to decrease systemic exposure by using local injections such as sub-Q administration, dose escalation, or delivery system that locally produce these bispecifics. And lastly, to alleviate the cytokine release syndrome, the studies in humanized mouse models have shown that blocking TNF-alpha and its downstream cytokines such as IL-6 can effectively mitigate CRS, and TNF-alpha produced by activated T cells leads to the secretion of inflammatory cytokines by monocytes. And these strategies together help make T-cell engagers more selective and specific for tumors and improve their efficacy while minimizing the side effects. To improve T-cell infiltration, pretreatment will generate a strong indifferent response in the tumor, and including local innate and adaptive immune responses and strong T-cell infiltration. And second, disruption of the extracellular matrix barrier by some components such as collagen can prevent T-cells from getting stuck in the ECM and allow infiltration into the tumors. And third, targeting the cancer-associated fibroblasts can remove the major producers of ECM components and immune-excluding cytokines such as CXCL12, and will lead to more T-cell infiltration and increased sensitivity to checkpoint inhibition therapy. Or to improve the quality of T-cell responses, we can target immune-suppressive factors such as immune-suppressive cells and cytokines, and thereby reducing the inhibitory signals to T-cells and improve their effector function. And second, considering the exhaustion and limited number of T-cells within the solid tumor. So combining CD3 bispecifics with other like checkpoint inhibitor therapy might enhance the response. Preclinical studies have shown that the combination therapy can enhance T-cell activation. And third, co-stimulatory signals can increase T-cell proliferation and thereby boosting the number of effector T-cells. Clinical model studies have shown that when used in combination with CD3 bispecifics, anti-4-1BB prolongs T-cell activation and enhances the anti-tumor efficacy. We know that preclinical evaluation is indispensable in the development of antibody drugs. Through rigorous preclinical evaluation, the risk of severe adverse reaction in clinical trials can be significantly reduced, thereby increasing the successful rates of drug development. In vitro, the specificity and affinity of the antibody target CD3 and the tumor-associated antigen will be analyzed. Methods including SPR, ELISA, or cell banding, and in vitro cytotoxicity will be conducted to measure the antibody's ability to mediate tumor cell lysis. T-cell activation and proliferation will also be evaluated to assess whether the antibody can effectively stimulate T-cell expansion and cytokine secretion. In vivo, the anti-tumor activity of the antibody will be tested in tumor-bearing mouse models to determine its efficacy in reducing the tumor size or extending the survival. And PK and PD studies are performed to understand the absorption, distribution, and mode of action of the antibody. Additionally, safety and toxicity are assessed through various animal models, focusing on potential impacts on vital organs like the liver, kidney, or heart, and to establish a safety dose. And tolerability, especially at higher dose, will be assessed to ensure the safety of the antibody's profile before going into a clinical stage. This is the Amgen's tarlatamab, which was granted accelerated approval by FDA this year. It is a DLL3 CD3 bispecific antibody for the treatment of extensive stage small cell lung cancer. Its AMG 757 is a half-life extended BiTE molecule designed to redirect T cells to tumor cells and induce T cell activation and expansion and killing of tumor cells. First, DLL3 specific activity of BiTE molecule was studied in vitro and in vivo for proof of concept, and the results demonstrated that the target is an optimal BiTE molecule target, and then the half-life extended drug AMG 757 was assessed in a target cell killing assay in vitro, and as the result shows, it can specifically mediate cytotoxicity against DLL3 positive cells, but not to the DLL3 negative cells, and It also exhibited dose-dependent cytotoxicity against different SCLC cell lines, including those with very low DLL expression levels. Here they analyzed T-cell activation and cytokine release in vitro to understand the mechanism of action of AMG 757. As we can see, the drug increased granzyme B levels and cytotoxicity over time. T-cell activation or inflammatory markers like CD69, CD71, PD1, and PD-L1 were upregulated following AMG 757 treatment. Tumor lysis was accompanied with the production of pro-inflammatory cytokines. These data indicated that AMG 757 dependent T-cell activation, cytokine production, and release of cytotoxic granules was associated with biomolecule target engagements. In vivo anti-tumor activity of AMG 757 were also evaluated in xenograft mouse models. As we can see here, the drug significantly inhibited the growth of established sub-Q tumors, or also topic lung tumors, and here the NCI-H82 metastatic liver lesion model. And both models used cell lines engineered to express luciferase to enable viralization of tumor burden. Here, a single dose of AMG 757 led to a statistically significant increase in the absolute number of human CD4 and CD8 T cells in the tumors. And the T cell activation markers CD25, CD69, and PD1 were significantly upregulated. Here is the IHC results, and demonstrated that CD4 and CD8 T cells were infiltrated. And all these results indicated that AMG 757 can promote T cell trafficking to tumors in different compartments and engage T cells to DLL expressing SCLC cells. In vivo PK and non-clinical safety assessment were conducted in non-human primates, and the AMG 757 is well tolerated in non-human primates. In toxicology studies, no AMG 757 related adverse finding up to the highest dose level tested. And the PK study shows that AMG 757 exhibits an extended half-life. Appropriate, stable, and reliable preclinical evaluation is crucial. At SMOKE, our industrial customer department offers a comprehensive range of pharmacology services for T-cell engager preclinical evaluation. Our services include in vitro pharmacology, in vivo PD evaluation, and toxicology analysis, and more. Considering the broad prospects of CD3 bispecifics or multispecific antibodies and the importance of preclinical in vivo drug evaluation, we have developed CD3EDG humanized mouse model. Why this model is a better choice for evaluate the drug candidates? The CD3 complex is one of the key molecules to modulate T-cell functions. The structure of the CD3 protein, including the N-terminal extracellular domain, a transmembrane domain, and a cytoplasmic tail region, contain the ITAM motif. And CD3 is composed of 4 different subunits. These subunits interact to form the CD3 complex, which associated with the alpha and beta chain of the TCR, together forming the complete TCR-CD3 complex. And the structural integrity of the TCR-CD3 complex is important for T-cell development and functional. Mouse is an excellent experimental animal. However, research into new bispecifics is hampered by the lack appropriates into in vivo mouse models due to poor cross-reactivity of human CD3 with mouse CD3. And one approach is humanize the mouse by recapturing the human immune system in immune-deficient mice. And these mice have a donor derived from human immune system that includes effector T cells. However, it is well known that several types of immune cells cannot be developed in these mice, so the adaptive immune function cannot be fully recovered. This model cannot be used to assess the combination therapy target, and this mouse has a donor-to-donor variability and GVHD. And another approach is to establish transgenic mice expressing the human CD3 complex. So here we developed a fully humanized CD3EDG mouse model for evaluating CD3 bispecific antibodies. This is a humanization strategy for CD3EDG humanized mouse. The mouse was generated in C57BL6 or bispecific background through stepwise targeting via ESL-based back transgene. And the mouse CD3 epsilon, delta, and gamma gene tandem, which is located at the same allele on chromosome 9, was completely replaced by human CD3-EDG gene tandem. First, we used homozygous humanized mice for further examination. According to the results, facts show that homozygous mice successfully express human CD3-EDG on T cell surface. We also characterized lymphocytes lineage development in the homozygous mouse, and as the result shows that the homozygous humanized CD3-EDG mouse have comparable levels of B cell development as wild-type mice. And the homozygous mice have comparable level of CD4, CD8 T cells as wild-type mice. And the homozygous mice also have the comparable levels of nature killer cells and Tregs as wild-type mice. And all these results indicated that our humanized CD3-EDG mice have a normal immune system and successfully express human CD3-EDG. We also carried out humanization on bispecific background and successfully obtained the humanized CD3-EDG mice. In the spleen of the mice, human CD3 epsilon and delta can be detected, and mouse CD3 expression as absent. In contrast, in wild-type control mice, only mouse CD3 can be detected. The result was also observed in blood samples of homozygous humanized mice, as we can see that human CD3 epsilon and gamma can only be detected in homozygous humanized mice, but not in wild-type control, and mouse CD3 expression is absent in peripheral blood of homozygous humanized mice. The function of T cells from this humanized mice was tested ex vivo. As we can see, isolated CD3 positive T cells from spleen showed normal activation and proliferation capability after stimulated with anti-CD3 CD28 comparable to the wild-type control. And the results indicated that this humanized CD3-EDG mouse in bispecific background have a normal T cell function. Next, the CD3 humanized mouse model was used to assess bispecific T cell engager. We engrafted MC38 human claudin 18.2 tumor cells in homozygous humanized mouse model, and we can see that at 1 MPK dosing group, the tumor growth inhibition was 83%, and in 0.3 MPK, the TGI is 63%. The homozygous humanized mice was also engrafted with MC38 human CEACM5, and after treatment with anti-CD3 CEACM5 bispecifics, the tumor showed a total regression. Another study using different anti-CD3 FOLR1 bispecific to treat homozygous humanized mice that were engrafted with MC38 human FOLR1, and all specific antibodies showed anti-tumor efficacy. So all these results indicated that our mouse model provide a predictive platform for in vivo efficacy of CD3 bispecific antibodies. Next, we use homozygous humanized mice to assess the anti-tumor response of anti-mouse PD-1. As we can see, the anti-mouse PD-1 antibody showed anti-tumor activity in a dose-dependent manner. And here, the tumor infiltration lymphocytes were examined by FACS. And we can see that anti-mouse PD-1 treatment induced the frequency of T cells. And this indicated that further combination therapy of CD3 bispecifics with the immune checkpoint inhibitor can be evaluated using the humanized CD3EDG mouse. Therapies involving T cell activation commonly accompanied by immune-related adverse effects, mainly the cytokine release syndrome. And this is initiated by T cell triggering, and monocytes and macrophages are the primary source of systemic toxic cytokine release. And by preventing TNF-alpha release, it is sufficient to impair the systemic release without affecting antitumor efficacy. And cytolytic activity of T cells can be achieved in the absence of cytokine release, and this mechanism is important for reducing the cytokine release side effects. In the following study, we will use homozygous humanized mouse to assess the in vivo AICD and IRAE assessments of bispecific T cell engager antibodies. To perform the assessment, homozygous humanized mouse was engrafted with MC38 human epicam tumor cells and treated with OKT3 or anti-CD3 epicam bispecific antibody at different time points. And according to the results, anti-CD3 epicam treatment lead to 83% tumor growth inhibition, while OKT3 treatment promoted tumor growth, likely due to the activation-induced cell death of T cells. Cytokine release was tested by ELISA. We can see that CRS was induced within 24 hours after OKT3 dosing, with L2, L6, and the TNF-alpha peaking 2 hours post the first dose, and interferon gamma, IL-10, and IL-5 peaking 6 hours post the first dose. In contrast, we can see no significant cytokine release was observed upon anti-CD3 human epicam treatment, despite the remarkable TGI that has already been shown. So, this delicate balance of in vivo T cell activation and T cell toxicity is crucial for effective tumor killing. And in another study, low dose and high dose levels of anti-CD3 epicam bispecifics were used to treat tumor-bearing humanized CD3EDG mouse. Both groups showed complete tumor regression, while anti-human CD3 negative control showed no efficacy. And we can see that no significant difference of cytokine release was observed among each group except L2 and interferon gamma. And these two cytokines indicate in vivo T cell activation. By crossing CD3EDG humanized mice with the variety of gene humanized mice that we already have, we have generated multiple gene humanized mice, including but not limited to the models that listed below in the table. In addition to CD3, we have humanized co-stimulator factors like 4-1BB and CD28, immune checkpoints like PD-L1, and also the tumor-associated antigens. For example, EpCAM, PSMA, or GPC3. All these models will allow us for a more accurate evaluation of CD3 bispecific, trispecific, or multispecific antibodies, and also combination therapies, such as with the immune checkpoint inhibitor. We have more than 500 gene humanized mice, and these models will allow us to generate more models for multispecific T-cell engager evaluation. Humanized cell lines are important for syngeneic tumor model development. We have more than 100 humanized cell lines for model development on CD3EDG humanized mice, which will allow us to evaluate T-cell engager with the different tumor-associated antigens. Well, to summarize, our CD3EDG humanized mouse model provides a predictive platform for in vivo efficacy and safety assessment of CD3 bispecific or multispecific antibodies, or even combination therapies. And this model also provides a biological rationale to clinically preventative treatment approaches to avoid or reduce CRS-related toxicity. When engrafted with a syngeneic murine cancer cell line, the in vivo efficacy testing will be performed in a fully immunocompetent environment. And furthermore, by cross the mouse with other humanized mouse strains, we can generate robust humanized target models. We offer a variety of mouse models, cell lines, and pharmacology and toxicology services to support the preclinical evaluation of CD3 bispecifics. If you have any questions, please feel free to contact us. Thank you.